When the progeny-tested plus trees have produced offspring, which has then been successfully initiated, proliferated, matured, germinated and grown in the nursery, it is time to plant the somatic seedlings. At the same time, the genotypes are cryopreserved to maintain their juvenility and capacity to multiply. The samples may be cryopreserved up to decades, for example until field testing results are available from the individual genotypes. In liquid nitrogen, the material is also very well protected from biotic factors such as diseases, storms and other major nature events. After propagation in the laboratory and growing in the nursery, the journey of Norway spruce SE plant continues to field testing. In the field, the plants are exposed to natural environment and their growth and development are carefully monitored. In this experimental site, 280 genotypes have been planted in same plots in 2014. In this kind of setup, it is easier to demonstrate the differences among families and genotypes. At this site, 5 years after planting, the plants of the fastest growing genotype were 74% taller than the mean of the full SIP families of progeny-tested plus trees. Among 60 and 120 fastest growing genotypes, the mean height was still 27 and 17% above the overall mean. Of course, growth is not the only factor for selection, but the growth differences are easy to measure and demonstrate visually. In Finland, field testing of Norway spruce SE material is carried out in a similar manner as other tree breeding material. In this test site planted in 2019, 160 genotypes from 12 full SIP families are grown together with control material, which are unimproved seedlings. The breeding value of the genotypes is tested on a series of sites such as this. The sites are located both inside and outside the utilization zone. Within the test sites, the plants have been randomized in several blocks where the development of individual plants is followed. Aforementioned 12 full SIP families have been registered as basic material for forest regeneration in 2017. The type of material is parents of families and the category is qualified. The national legislation in Finland, derived from the EU directive, sets production limits to this material. This test site planted in 2018 serves both research and process development. Besides testing the breeding properties, it is necessary to test how the treatments carried out in the laboratory and nursery affect to the plants after planting on site. SE plants growing on this site were transplanted directly to nursery after 1, 3 or 5 weeks of in vitro germination after varying cold storage treatments. The treatments were tested on 22 genotypes from 12 full SIP families and SE plants from all genotypes and treatments are planted on this site. Most of the plants at this site were germinated only for 1 week in vitro because when combined with a suitable cold storage treatment it proved to be most beneficial for survival of SE plants.